Hello, everybody. This is George Yancopoulos, and I want to welcome you to this morning's panel on immunology from both sides, oncology and autoimmune disease, on which I'm joined by Carl June and Nancy Simonian. Before we get to our regularly scheduled topic, we all know that the COVID-19 pandemic is at the top of everybody's mind. You all just heard from Tony Fauci, where he told you all the sobering facts, but he also referred to the fact that he's cautiously optimistic on the promise of monoclonal antibodies. And many of you are probably aware of our recent results with our anti-COVID-19 antibody cocktail treatment. That just last night, we released overwhelmingly positive results from our confirmatory study in COVID-19 outpatients. So before our regular topic, I was asked to provide a brief overview for everyone, about 5 minutes, about these results. And then we'll go to the regularly scheduled panel. So just to provide some background, our Regen-CoV-2 cocktail, that's what we call it, consists of 2 potent neutralizing antibodies targeting distinct epitopes on the critical receptor binding domain of this spike protein that you already heard about from Dr. Fauci. It's important to point out that we previously used an analogous approach to develop a similar cocktail to target the Ebola virus. And our Regen EB3 cocktail was just approved last week by the FDA as the first approved treatment for Ebola based on providing a significant mortality benefit as published in the New England Journal of Medicine. So obviously our success with Ebola created a lot of hope for our very similar approach against COVID-19. Now, the use of multiple antibodies in our cocktail was important, and we showed that it was important to prevent so-called mutational escape by the virus. We demonstrated this in preclinical experiments as compared to single antibody treatments. And preventing escape mitigates against loss of efficacy, and perhaps most importantly, prevents the seeding of viral escape mutants into the broader population. And unfortunately, if you seed such escape mutants, that might create resistance not only to other antibody treatments, but even to vaccines more broadly. So the antiviral activity of our cocktail has been validated in an assortment of preclinical studies showing viral load reductions in both the prophylactic and therapeutic setting in non-human primates. Right now, our cocktail is in multiple clinical trials, both as a prophylaxis in people at high risk of contracting COVID-19, in early patients to prevent progression to more serious disease and to hospitalization, as well as trials in advanced hospitalized patients. Just a few weeks ago, we reported the initial descriptive results from our study of 275 mild to moderate outpatients, showing potent antiviral activity and also associated clinical benefit of reducing the need in these mild to moderate patients for further medical attention. So the recent excitement was based that just yesterday, last night, we released prospective and overwhelmingly positive results from a larger analysis now involving 524 additional patients, confirming the antiviral activity, as well as demonstrating a statistically significant ability to prevent the need for further medical attention, such as hospitalizations and ER visit. And most importantly, as you just heard about the individuals who are at highest risk, the benefits seem to be most pronounced in those who are at highest risks. So obviously, we're all been waiting for promising and exciting news against this horrific and tragic pandemic. And we do think this news and this data and this cocktail provides such potential and such promise. And we're in the process of discussing an emergency use authorization for this cocktail with the FDA. You've already heard from Tony Fauci. You'll be hearing a lot more about vaccines throughout the day. We need obviously multiple weapons. There's going to be roles for all of these in our attempts to fight back and end this pandemic. And so I think that there is a lot of reason for hope. and we're excited about these possibilities. And we really hope that all together, our scientific community can continue to bring promise and potential against the pandemic. So with that brief summary, I want to get back to the subject of our panel. And I want to start by first just letting you know that there are going to be on-screen polls that you can, in live form, respond to and give your perspectives. And also, there's also an on-screen Q&A. If you write in the Q&A and your name and your question, I will then direct those to the panelists as appropriate. So I'm going to start by introducing Carl June and Nancy Simonian. Carl is the Richard W. Vague Professor in Immunotherapy, the Director of the Center for Cellular Immunotherapies, and the Director of the Parker Institute for Cancer Immunotherapy in the Perelman School of Medicine at the University of Pennsylvania. Carl has won numerous major awards and is widely recognized for his pioneering work on so-called CAR T therapy, in which patients with refractory and relapsed chronic lymphocytic leukemia were treated with genetically engineered versions of their own T cells, designed to target and kill the leukemia cells. Carl's work has been widely recognized as a major turning point that is delivering on the long-held promise of cancer gene and immunotherapy. The CAR T cells invented by Dr. June's laboratory were awarded breakthrough status by the FDA in 2014 and ultimately approved by the FDA for acute leukemia in 2017 and shortly afterwards for diffuse large B-cell lymphoma. Carl is truly someone who, with his science, is changing our world. Nancy is the president and founding CEO of Syros and has an established track record of value creation in biotechnology. She has led the evolution of Syros from its inception into a publicly traded company with 2 drug candidates in clinical trials, a rich preclinical pipeline, and a leading gene control platform. Prior to Syros, she was the chief medical officer at Millennium and previously vice president of clinical development at Biogen. Most notably, under Nancy's leadership at Millennium, Velcade became a mainstay of treatment for multiple myeloma. At Biogen, Nancy played a central role in developing treatments for multiple sclerosis. Based on having these rare successes in developing treatments for both cancer and autoimmune diseases, Nancy is the perfect individual to discuss the intersections between cancer and autoimmune diseases today. So now that we know a little bit about our distinguished panelists, I want to start first with Carl, asking for his thoughts based on being a pioneer and an expert in CAR T immunotherapy against cancer, and on having now real-life experiences on how various immunotherapies against cancer can result in excess immune activation, ranging from cytokine release syndrome to autoimmune diseases, from the more common thyroiditis and diabetes that is seen to rare life-threatening conditions like pneumonitis, and what he thinks the implications of learning are, and whether he thinks these learnings can be used to, first, better target these therapies to avoid autoimmune sequelae, and second, is it possible to take advantage of these learnings and reverse engineer some of these approaches and some of these targets so as to control autoimmune diseases? So, Carl?
Thanks very much, George, for that very nice introduction. And I want to thank you and your team at Regeneron for what you've done, as you just mentioned, with antibody therapy. So in the old immunology terms, we would call that serotherapy or passive immunization. You can give antibodies to someone and instantly they can be protected. That's similar to when our immune system, how it works. For instance, when a baby's born, they get their initial immunity from their mother's breast milk. What I've worked on is in between a vaccine therapy where you have to generate your own immune response, and that's where your own immune, and that takes time. With cell therapy, we transfer, and it's a form of passive therapy, but it's not made by antibodies. It's made by the T cells that go in, and then they can be instantly on patrol for infected cells, such as a viral cell or tumor cells. So it is a form of passive therapy, but the cells stay in and then they can divide in the patient. And our first patient we treated in 2010, just in August, he came back in the middle of our COVID pandemic, and we found he still has no leukemia. He's probably cured at this point. We can say it's 10 years on, but he still has those CAR T cells in him. So we know that they can survive for at least a decade in a patient. And so we have many, fortunately, kinds of immunotherapy we can do now, ranging from vaccines that we're also interested to hear about to kinds of passive immunotherapy, antibody transfers such as your cocktail at Regeneron, and our kinds of CAR T cells that we make for leukemia. Now, the sad thing is when I went to medical school and you did, we didn't know what caused cancer. We thought it was viruses mostly, and it turns out maybe viruses cause only 10 or 20% of human cancer. The rest is caused by mutations. And we've learned that in great detail now, and it's given us new kinds of therapies. And in this, we still don't know many of the causes of autoimmune disease. That is, I think, a paradox that we've really solved what caused cancer. Now, the therapies are very difficult and they're happening, but autoimmune disease remains, in many cases, unknown the cause. Now, in 2011, checkpoint therapies got approved. Those are the antibodies that Bristol Myers sold for metastatic melanoma and now many different cancers. And we found that some people got autoimmune disease after being treated, and they call these immune-related adverse events. And the immune system is such a potent probe, and very potent. One cell can reveal things. What we found out now is new things that were completely missed for decades. So in one case, we find some of the autoimmune side effects are it attacks your pituitary gland. And that leads to a variety of issues that if they're not treated, it can be fatal without your pituitary gland that makes all our hormones. And what they found was completely missed for decades by all the anatomic studies is the pituitary gland expresses CTLA-4, which is targeted by the drug ipilimumab for melanoma. So we've learned something very new. Just last month, my group published in Cell that the brain expresses the molecule CD19, which is the target of our CAR cells, on cells that line blood vessels in the brain. Completely missed, for ever since monoclonal antibodies were invented in 1979, no one thought that molecule CD19 was anywhere other than in a B cell and in leukemia cells. So this, intersection of autoimmunity and cancer is teaching us a lot of underlying mechanisms now that were completely unexpected before. And that one will allow us to sharpen the sword so that we can avoid those. But that's just two examples, CD19 in the brain and CTLA-4 in the pituitary gland in the brain. And it's only now because we get these very active T cells that we can discover these rare populations of cells.
Well, Carl, do you think, obviously, including now with CAR T therapies, you and others, but also the immunotherapies that you mentioned from BMS and others, are trying to block these immune regulators that keep the immune system under control. And by releasing these breaks from the immune system that normally operate, it unleashes more activity, which can kill cancer cells, but like you just said, it can also kill the pituitary, or it can attack the lining of vessels lining the brain, and so forth and others. Do you think that the learnings that we have here might teach us how to do the opposite, that increase the break, further stop the immune system when it's overactivating, resulting in autoimmune disease? And do you think those are exciting, valuable approaches, or are they going to lead to more problems?
Yeah, I think I think without a doubt, we're going to find ways now to both dial up the immune system, which is what we want to do to control infections and cancer, or turn it down in disease. And the two major areas there are organ transplantation. So if you get a kidney transplant right now, most people have to take drugs to suppress the entire immune system for the rest of their life. And that's the central problem that's been called the holy grail of immunology, is how do you induce tolerance so that you can now, say, put an organ in from someone else, unless you have an identical twin. And there are even companies now making, for instance, inbred pigs and other animals that will grow organs, a replacement heart or kidney. But the real issue then, they work. The issue is making it so your immune system doesn't reject that. And there are now, there's been the appreciation, again, since I was in medical school, that there's very sophisticated ways to induce tolerance of your immune system. And so tolerance means that your immune system will ignore that. It could be an organ, such as a pancreas, or it could be certain kinds of cells. This tolerance then means it leaves them alone, but yet the immune system isn't suppressed, and it can still work to get rid of invaders, such as a new infection like Ebola, or it can, and so stay on the job without being, suppressed entirely, which is what we do when we take like high dose steroids and so on. They're in the immune system, you're left wide open to all kinds of infections, and probably getting cancer as well, which we could talk about. So getting this induction of tolerance has been the holy grail. And we found out that the major cell that does that in the immune system is called a regulatory T cell, or Treg. And there are now trials around the country making CAR Tregs, so they can put, genetically modify these Tregs. And what they do is jack down the immune system to, for instance, a specific organ. One area that's being tested is, could you take autoimmune diabetes, where people have to take insulin all their life and they lose the islet cells that make insulin? Could you shut that off by giving cells that suppress the body's immune reaction to the pancreas? And then maybe you could give them some more islets and they would be cured of diabetes. That's just one example.
Well, do you think, obviously, one of the most effective targets, one of the most effective breaks that is being blocked with immunotherapies is PD-1, which unleashes more killer T cell activity against the cancer. Do you think it's possible that it could be as simple as adjusting the tone of PD-1, that is, in people who are suffering from autoimmune disease, or in the case of transplant, instead of completely paralyzing the T cells, as you said, with some of the T cell toxins? Could you imagine where one literally just gives almost a general agonist of PD-1 to just do the reverse of what we do in cancer, activate the PD-1, and in this way actually get a little bit more turn off, or a little bit more? By activating PD-1, you're turning off the T cell a little bit.
Yeah, that's going to be one of the general strategies. So Ron Evans at the Salk Institute recently had a paper in Nature doing exactly that. So he made, I think, from induced pluripotent stem cells, he could make islet cells that make insulin, but he put in there, engineered in genetically, PD-L1, and then showed that this is all in mice, it's not close to human trials, but then it would induce tolerance. So that's dialing up and dialing down, as you said, these checkpoints. But it's very complicated, unfortunately. It's not going to be like one shoe size fits all. So it's turned out the worst autoimmune side effect from PD-1 is myocarditis. It's rare, but when it happens, the immune system gets targeted against the heart with PD-1 checkpoint therapy, and half the people die. I mean, it's awful when it happens. The immune system literally can destroy your heart in a matter of a week. And it turns out the heart uses, as a special way to protect itself, PD-L1. Now, I just mentioned our pituitary gland uses a different one, which is CTLA-4. So there's organ-specific toxicities, and it would need to be taken into account. you certainly don't want to take away the heart's protection from being auto-attacked in autoimmune disease, which is, it's a PD-1, PD-L1 based system. So I think, it will be doable. Some of these things are going to be personalized because it depends on people's own background. Like some people have family histories of autoimmune disease. It's common, like for instance, if you have type 1 diabetes and you have an identical twin, they have up to a 50% chance of getting diabetes themselves. So it's genetic. In other cases, it's infection related. But I do think we're going to be able to tune up and tune down things like that to treat both organ rejection and autoimmunity, much as we're doing with cancer now.
All right, thanks, Carl. We're going to come back to you, but let's start now with Nancy. Nancy, you're an expert in understanding gene regulation. Your company is focusing on small molecule drugs that can actually mediate gene regulation in useful ways in various applications. Do you want to comment on your learnings of genetic pathways and so forth? Are there new pathways? Are there approaches, either on the tumor side or on the immune cell side, which you think might be useful in either direction for either dialing up the immune system in the case of cancer or dialing it down for autoimmune disease? And with all your experience in cancer and autoimmune therapies, what do you think about the intersection of therapies there and the implications going in both directions as well?
Yeah, thanks, George. And first, congrats on the great progress on the COVID front. It's so important for all of us. I'm going to approach my comments as someone who is trained as a physician and has been involved in drug development in both cancer and immune-mediated conditions. And I would say that, in my opinion, the most important thing we can do to combat autoimmune diseases and cancer is to understand the fundamental disease biology that causes or contributes to the disease. And I believe that's actually where we have the greatest need. I'm convinced that if we find the right target and the right pathways for the right patient, we can develop ways to modulate. So it really comes down to the biology and the patient. And just stepping back, one of the best ways to really understand disease biology, human disease biology, is human genetics. And from a drug discovery and development standpoint, you have a very high degree of confidence when you have a human genetic experiment where you have a mutation in a protein and that mutation causes a disease. You have a high degree of confidence that you know where to go. And how has that been applied to cancer and autoimmune diseases? Well, in cancer, as George said, Carl said, it is a genetic disease. And we certainly have had some success by targeting mutations in protein coding regions. But for the most part, those have been sort of relatively short-lived in terms of the clinical benefit. And that's because there's a tremendous amount of heterogeneity. In a tumor, every single cell is different in terms of its mutation landscape or its function and whether it's more like a stem cell or more likely to metastasize. And tumors get around kind of going after sort of single mutational changes. So while genetics has been very important and has led to a lot of important treatments, it hasn't solving really the issue that we have with heterogeneity. And also we know that cancer and the manifestations of cancer is a result of a lot of non-mutated cells, the immune cells, the endothelial cells, an exhausted T cell. How's that different than one that's stimulated? That's not related to mutational changes, it's functional changes. And we don't have a good handle on that from just pure human genetics. And then if you turn to autoimmune diseases, it has not been thought about as a traditionally genetic disease. The immune cells that are dysfunctional are mutated. Their function is altered to attack normal cells, and we don't really understand all the basis for that. So I would sort of step back and say, to combat those diseases further, we have the basic challenges as if we don't right now have all the tools and lenses that we need to understand that. And then secondly, I think the challenge, but both the opportunity is now that we're perturbing all these diseases with drugs in the clinic, there's so much that we can learn further from the patient experience or the patient experiment. And so I want to touch on both of those. So on the first, in terms of the lens, one of the things that we now have the ability to do really over the last several years based on sort of new technologies and DNA sequencing costs coming down, and is to interrogate sort of the gene program that regulates the function of cells. And there's specific regions in cells that are called enhancers. There's those switches that turn up or down the expression of genes. And the very simple way to think about it is, you have hundreds of different cell types in your body, a skin cell or a muscle cell. They have identical DNA. How they differ is in their function because of the program of genes that are expressed. And we now have the ability to understand that in an unprecedented way. And we can also understand that in terms of a cell functioning in different ways and what is different at the molecular basis. We also know from GWAS studies, now this is related to autoimmune diseases, where we haven't been able to find mutations in protein coding regions that causes diseases, but that the greatest amount of human genetic variation that's associated with autoimmune diseases are in these enhancer regions or in these gene control regions, which means sort of the altered expression of a gene, either a little bit too much or too little, predisposes people to autoimmune diseases. So how can we apply these things to think about a better understanding of disease biology? So I'm going to give you an example of how we've applied it at Syros, my company. Using these new technologies, we were able to analyze primary human leukemia cells, focusing on these switch regions of the genome, and identified that about a third of patients with acute myelogenous leukemia had an alteration in the regulation of a gene. And when that gene was altered, the expression of it, those cells became dependent on a pathway that then we were able to target with the drug. And today we have data in the clinic by targeting that molecular subset based on its gene regulation pattern, that we, in combination with a standard of care drug, could put 60% of those patients into a complete remission with that combination. So I'm really excited about the potential of sort of a new ability to understand the function or dysfunction of disease cells and use that understanding for new therapy. I think along those lines, I think one of the most important advancement that's coming along, or I call it refinement, is I mentioned about the heterogeneity. So we used to kind of look at a tumor and think, well, a lot of these cells are the same and we can just look at them in bulk. But as I mentioned before, I think we now know that the tumor itself is very heterogeneous, and also all the surrounding cells, the immune cells, are not all in the same state. So now there's a new technology called single cell RNA-seq. Essentially, it allows you to kind of profile single cells for their function. And therefore you can show that, are tumors enriched for cells with a certain functional type? Are immune cells within a tumor, what is their functional type and how do they differ? And why that's so important is we're now learning that depending on what we understand about the function of individual cells, certain treatments may work or not work. And as an example, a recent paper, again, related to AML, showed that by using single cell RNA-seq, that the 30% of patients that were resistant to a standard of care drug all had the functional function of their tumor cells quite different. They looked much more like mature monocytes than stem cells. So already now we can sort of say, well, what's different about those cells, those function of those cells, that we now know we can come in and target that for a resistant population of tumors. So I think this holds a lot of promise, the ability to functional assess at a single cell level to better combat cancer. So how can we apply these to immune mediated diseases or immune cells in cancer? Well, we can do exactly the same thing for immune cells. And it's actually probably even more important there because there's no genetic alterations. So we've applied that to looking at macrophages coming from tumor tissue, looking at immunostimulatory macrophages or immunosuppressed macrophages. And we've been able to show differences in the gene control program between the two. We've actually been able to identify targets that are important in that altered function of one or the other. And interestingly, when we then in animal models, when one knocks down that target or pathway, it actually results in autoimmune disease. So I think by going after and understanding the immune cells in cancer and what may be important in stimulating it, we're already gaining insights into how that may be applied to autoimmune diseases. So I think that the only other thing I want to say, it relates to autoimmune diseases that I think has been a real challenge is, today in cancer field, we can now say, what type of cancer do you have? You have this histology, so we use breast cancer. Now, you don't ask just you don't ask what kind of cancer, is it breast cancer? You say, well, what type of breast cancer is it? Does it overexpress estrogen receptor? Is it HER2 positive? And not just that, now you're going to the next level. You're saying what's altered at the mutational level within that subtype. And that's giving us a lot of ability to understand how best to treat a patient. We do not have the same ability to do that in autoimmune diseases. My training, medical training was as a neurologist, and even today, when someone comes in and presents with multiple sclerosis, we don't have the ability to really understand how one patient differs from another. We're still characterizing those diseases by clinical course. I think the more that we can use some of these functional technologies to understand the differences, because MS or RA or IBD are not homogeneous diseases, but we are really behind in our ability to understand them at the individual patient level. So the last comment that I want to make, and I think you heard a little bit about this from Carl already, is, I mentioned up front the challenges that we have in trying to combat these diseases. And so, using better approaches to understand function. But I also think that what we learn in the clinic is going to be so important, and we need to do more and everything we can, especially with new therapies coming up for cancer, like learning as much as we can about the human disease situation to be relevant. So what do we know about the immune system and cancer and autoimmunity? And I think I would just sort of step back and just say, it's very clear that kind of balance in the ability to modulate, I think you heard that word from George, is really important, because too much or too little of a good thing can be a bad thing. And I think that's the sort of the underlying premise when you think about immunology. So obviously too much inflammation isn't good. So we know that autoimmune diseases predispose people to cancer. So if you have inflammatory bowel disease, you have an increased risk of colorectal cancer. If you have RA, you have an increased risk of lung cancer. And that's probably a result of sort of the genotoxic stress of immune cells on a tissue. So too much is not good. On the other hand, too little inflammation is also not good. We know that drugs that dampen the immune system can increase the risk of cancer or oftentimes secondary cancers. Immunosurveillance becomes compromised, so you don't want too little. We know that some of the drugs used to treat cancer can help people with autoimmune diseases. Methotrexate, other immunosuppressant drugs like steroids, they can be used in both diseases. And then finally, as we're learning now, some drugs to treat cancer, like checkpoint inhibitors, can cause autoimmunity as an adverse event. And there's so much, and you heard from Carl, that we're learning about that can help us. So I would just say that there is a rich source and a growing rich source of human data and experience that we have and that will grow, and that we should leverage those findings to help us think about new ways to combat autoimmune diseases.
Well, that was fascinating. It actually reminds me this intersection and the roles of immunosurveillance and cancer and so forth. Certainly remind me of a story that is one that's near and dear to us, that was really developed here at Regeneron, led in our group by Izzy Lowy and Matt Fury, which I think highlights this point about immunosurveillance and cancer, which is that a number of years ago, when we were looking for applications for our PD-1 antibody that inhibits or blocks PD-1 to help activate the immune system by unleashing the brake, I think they had a very unique approach that I was actually shocked and surprised that nobody else had seemed to have focused on, though both of you touched on this precedent, which is that we looked around and we said, well, if the immune system is normally playing a surveillance role, then classically immunosuppressed individuals, like organ transplants people like Carl was talking about, should then have dramatic increases in cancer. It turns out that they don't really, at least in terms of solid cancers, have as much of an increase in solid cancers as you might expect if the at least T cell wing of the immune system was playing such a critical immunosurveillance role, except for some limited kinds of cancer. And what Izzy and Matt realized was that the most dramatically induced cancer in the setting of classic immunosuppression was cutaneous squamous cell carcinoma, and that it was induced about 200-fold in immunosuppressed individuals. So they reasoned, which I thought was pretty brilliant, that if something that would help the immune system and hype it up, if you wanted to test such an agent, perhaps it would have the best activity against cancers that were normally controlled by the immune system and by immunosurveillance. So they went out and they were the first people to try a PD-1 blocker in cutaneous squamous cell cancer, which, as you know, is one of the most common cancers in the world. Fortunately, something like 95 to 98% of the cases can be successfully treated surgically. What people unfortunately ignore much of the time is, even because there's 1 to 2 million cases just in the United States each year, a couple percent progressing past that then become incurable. And there was no treatments for advanced cutaneous squamous cell carcinoma, and there's 10,000 or so cases of advanced disease just in the United States alone. Anyway, they tried our PD-1 antibody there, and they had spectacular success. Actually, probably for solid cancer, probably among the highest response and control rates ever described, showing that the immune system, for at least some cancers, definitively plays immunosurveillance role. That was known for CSCC based on what you saw in solid organ transplant patients. But then you could now, in non-immunosuppressed people, you could hype up the immune system by giving PD-1 and have dramatic results on this cancer alone. Do you guys have any thoughts on that, or how to take advantage of that? And what the learnings might be in that, the vast majority of people who get cutaneous squamous cell carcinoma aren't known to be immunosuppressed. But do they have some sort of deficits in their immune system that hasn't been studied, that maybe your approaches, for example, could find? And so when we are sort of non-specifically hyping up the immune system by blocking the brake, are there maybe other ways? And are there specific things that that people might be suffering from in terms of immunosuppression that are leading to these cancers? And maybe there's similar stories out there that other people haven't considered. So what do you guys think about that?
So I myself have had 6 squamous cell cancers that you're discussing, and surgery up here. And so we know that in part it's related to environment, UV damage to DNA, sun exposure, like melanoma. Most of them have KRAS mutations, a lot of them do. So similar to the awful tumors that kill us in the, for instance, pancreas. And it's what you said, if you get it out early surgically, you're cured, but then once it metastasizes, it's really bad. The major cancers are of the skin, it's melanoma, which happens in young and old, and that's sun exposure, skin. It was squamous cell is mostly, it's the older you get, the higher. And then there's another one that's emerging, which is called Merkel cell carcinoma of the skin. And Merkel cell, it was completely, my college roommate got it and he had to have his leg amputated. It turns out it's caused by a virus, a polyoma virus. And then that has antigens, and their checkpoint therapy probably with your antibody would probably work there. That's rare compared to squamous cell. So, the skin has a variety of cancers. It's also a site where our immune system has surveillance, where invasive organisms can enter. It's either you breathe them in like COVID, or they get on the skin and then you get an infection through the skin. And so those portals like that have different sensing things to set off the alarm. And in the skin, it's certain cells called Langerhans dendritic cells and so on. And they're looking for mutations, for instance, in your skin that would lead to a cancer. And you can jack that up with a reagent like your PD-1 antagonist. So you can increase immune surveillance in the skin. But then, overactive, about 10% of humans have overactive skin hyperproliferation that gives psoriasis. So a side effect could be something like that, this balance of having, that's an autoimmune issue with the skin, is psoriasis. And that's treated then with immunosuppression. So we're at the area now where, thanks to these amazing changes with cell engineering, protein engineering, and so on, we can target things, almost anything imaginable. And now these technologies, like Nancy mentioned, single cell RNA-seq is a revolution. It's like, that's how we accidentally discovered CD19 in the brain, and now the cause of one of the side effects of CAR T cells. And that was only because of single cell RNA-seq. So I think we're at a point where, unfortunately, it depends on your own genetic background as where your Achilles heel is, and some people it's going to be, they're predisposed to get MS or psoriasis, and others they're predisposed to get cancer. So we know that, familial breast and ovarian cancer, that's a horrendous thing. Well, and that runs in families. We know it's usually due to DNA repair defects. But I'm wondering, I think, where we'll get is immunoprevention there. Right now, it's these Angelina Jolie things, where people have mastectomies and their ovaries removed once they've had a close family relative. But I'm hoping we'll get to a point where we could use these immune modulators and then, use immunosurveillance to cull out tumors early. So, and that would be one obvious thing. You could identify people at risk for getting cancer by genetics. In this case, breast and ovarian. It also turns out some men will be at increased risk of pancreatic cancer if they have BRCA1 deficiency. So hopefully we'll get to a point of preventing cancer that way, and identifying early so-called approaches like that, either by looking for circulating tumor DNA that's, or family history. It's turned out, we haven't talked much, that some autoimmune disease is caused by immunosurveillance. And there's been a disease called dermatomyositis, which occurs generally in older people. And when you look a subset of those people, you'll find that they have an undiagnosed cancer. And the immune system, in trying to get rid of the cancer, has now caused an autoimmune disease against the muscles and skin. Same thing in some women with ovarian cancer. You'll sometimes find them, they come in with cerebellar problems. And the problem is that they have an ovarian cancer that's caused loss of tolerance in the brain. And so that could be a good thing if the tumor is still resectable. But once you've started autoimmune disease, it's really hard to stop it. But and it's Hopkins group in the last couple of years has really great data that scleroderma, which is one of the worst autoimmune diseases that happens in the skin, is caused by mutations, and then the immune system trying to get, surveilling out the cancer, it triggers this awful autoimmune disease. So, we're a lot more complex than mice. If you have inbred mice, it's been pretty easy to figure out what leads to diabetes. Type, NOD mice, they get it, and you can cure NOD mice quite easily, but humans are much more complex, the immune system and its interplay with these different organs. But I think the good thing for everyone to understand here today is that the technologies are finally here now to solve that complexity. And but a lot of it's going to be precision-based and individual. That's what Nancy said, and it's based on our own genetic backgrounds. We each have our own, Achilles heels.
Well, Nancy, do you have some thoughts on exactly what Carl is talking about? And, let me just bring it back to the example of some of these cancers that are clearly increased in immunosuppressed individuals. When they come up, when the same cancers come up, like Carl was saying, skin cancers are due to multifactorial mutations and so forth. Now, is it possible that the non-classical immunosuppressed individuals who get them, like Carl, have some small hole that we haven't recognized, that maybe your gene regulation approaches and RNA-seq approaches can find in such individuals, and those can be new classes of pathways and targets for for both cancer and for controlling autoimmune disease?
Yeah, no, I definitely think so. As I said, I think there's so much we can learn from the experience of people. Like so people on the same, drugs, why do some develop a cancer and why do not? Or why do some develop an autoimmune disease, and what's different about them? The more that we can understand those differences, that will give us a clue to, and it may be that, each of our immune systems is tuned a little bit differently. It's kind of how we're wired, and we may hyper respond to something or not respond enough. My guess is that it's related to that, and that then determines how we respond to a drug, whether we're more likely to develop a cancer or an autoimmune disease. And I think we just need to, and the only way we're going to figure this out is to study it in people, because as Carl said, the models are just too simplistic. And I think the more that we can understand from the human experience and mine everything we can about what people actually experience and what people develop with what drugs is going to be really key to that.
Okay, we have time for a couple questions. If you have some questions, you can put them into the Q&A, type them in with your name and association. We'll be glad to ask our panelists. The first question is from Scott Chandler, and I'll address it to Carl because he already mentioned regulatory T cells. And the question is about the regulatory T cells and the concept of exhaustion. I myself am not very happy with the term or the concept of exhaustion and so forth. I consider it more a concept of really regulation. But what do you think, Carl, about just the concept of regulatory T cells impacting sort of the balance and the concept of exhaustion?
Yeah, so, okay, so regulatory T cells, as I mentioned, are things that dampen down and they can do it in an organ-specific way. And so the immune response, and there are kind of two kinds of regulatory T cells, one that's made in your thymus from an original developmental decision to be a cell that suppresses the immune response. And then there's also a concept called bystander suppression, where in the periphery, T cells can be tuned either to be where they will be effector cells and kill virally infected cells, or they'll shut down the immune system, depending on, for instance, growth factors like TGF-beta or IL-6. So it's a complex balance of how much T regulatory cells you have and how many killer cells you have that kind of set us whether we're set up to get autoimmune disease, or because you don't have enough of these regulatory cells, or you're immunosuppressed and you probably get cancer outgrowth and other side effects such as bacterial infections. So the concept of exhaustion is relatively new. And it used to be thought cells would divide and then you could have T cells the rest of your life, and that they would sometimes become senescent, which means they're kind of locked in G1 phase. They can't divide anymore, but they just hang around and take up space. Exhausted cells now have been discovered, and they're epigenetically reprogrammed. So they're, and to what they do is then they're still able to do some functions, but they're kind of worn out. They don't have the ability to proliferate a lot anymore like new healthy T cells. And these exhausted cells, so it turns out cancer promotes these dysfunctional or exhausted cells as a way to hide. And that's one of the central problems we have in cancer immunology is fixing this problem of exhaustion. Now, your PD-1 antagonist pathway drugs that you make with Regeneron in part do that. They partly overcome exhaustion, but they don't fix it. There is this epigenetic rewiring, which is where a lot of people are working on, but it's not a solved problem yet. Now, so you can, where it's first been realized is exhaustion in our effector cells. And then that means your cancer progresses or infections such as hepatitis and so on progress, and you lose control of them. And there's also this concept now that Scott Chandler is asking about, which is in Tregs, do they get exhausted? And that may then lead, because if they get hypofunction, to the emergence of autoimmune disease. What we know is that with age, your immune system flags. In general, even a response to a standard flu vaccine in a 60-year-old, only half of us respond. Whereas if you're 30, everyone responds. So that's immune senescence. And then in some people, this exhaustion issue happens more in one arm of the immune response, the Tregs, than the other. And there's, as the question asks, there's complex regulation with such as things as neuropeptides. And so that we really don't know yet. That, it used to be thought there were just Th1 cells, which, kill cancer cells and viruses, and Th2 cells, which are used for allergy and worms. And now it's known to be much more complex. And these exhausted cells have just kind of entered the arena and are really, there's a lot of discovery going on in that area.
Yeah, I personally don't like the term exhaustion. I think it's regulated in some fashion.
Yeah.
But turning to another question, maybe one for Nancy, from Sam Brower. We just heard from Carl that he thinks that mice are too simple a model and not complex enough to reflect everything that's going on in humans. But Sam is asking about organoid models for both cancer and somehow trying to mix it and reproduce the immune system in there. If mice are not enough, is it possible to really believe that organoids are going to help and solve it? Or do we really need, as you were saying, to focus more on humans and trying to learn the lessons there?
Yeah, it's a great question. Let's just think a little bit about kind of evolution of cancer models. So first it was mainly cell line derived models where we, cured a lot of mice with those cell line derived tumors, but didn't really translate into the clinic very frequently. Then we moved, and we've been moving, over the last, decade or so to, patient-derived xenograft models. So we can take a tumor from a patient. Obviously, there's heterogeneity in that tumor. They go into mice. They don't all take, we're not quite sure that the exact heterogeneity that's in the patient remains in the mouse, but it is certainly more human-like. I think that's been very helpful. I think there's data that suggests that those models have been more predictive of what happens in the clinic, but they're still not that predictive. Of course, those current mice models don't have an immune system. And I know that there are models now being generated and being used where you can combine both the human tumor with the human immune system. I think, just on face value, that seems like that could lend better information than we can with the two previous ones. And, as we talked about before, the cancer is not just the tumor cells, it's everything around the tumor cells. So the ability to assay and understand the whole complex of the tumor and the non-tumor cells is really important. Ideally, yes, we would do everything in people, but if you're developing or discovering drugs, you got to have some model systems by which you can determine what you want to take forward or not take forward and what you think makes sense. So I do think that the organoid models, I think that refinement of being more the complexity that actually exists in people is a good step forward to better understand. Now, how that will exactly translate, we're gonna learn. But I think all of these model refinements are getting us closer, never replacing the actual human experiment, but the reality is that we can't discover drugs and figure out and validate targets only in people. It just is not possible. We have to make decisions. And so we need these models to make decisions.
Okay, last couple of minutes here, rapid fire. We'll have one question for each of you. Just, 1 to 2 minutes, just comment. Question on Scott Chandler. What do you think about the intersections between cancer and neurodegenerative disease? Are there any relationships there that you would see or want to comment, Carl?
I'm not a neuroscientist, and Nancy is. What I can say is, there's definitely inflammation in many conditions of these neurodegenerative diseases. So I think that's part of the equation. And whether manipulating cells in the brain to change the immune balance might well change. We've actually made some cells to try to phagocytose and take up neurofibrillary tangles and so on, and plaques in mice. We're doing that here with Virginia and John Trojanowski here at Penn. But I don't know. I think clearly inflammation is important, and I'll stop there.
Okay. All right, Nancy, for you. Do you want to make some comment about the microbiome? Is there a big story there that we're missing in terms of the immune system interactions, both for autoimmune disease and cancer. You got the last minute is yours there, Nancy, on that.
Well, I mean, I think there's some really intriguing data that links the microbiome to, response to, checkpoint inhibitors and other drugs. There clearly is evidence that, changes that happen in the gut have effects on immune cells. So I think it's a really interesting area. I think we don't have all the basic tools and technologies and mechanisms to fully understand that, but I think there's a lot of intriguing evidence that there is a link there. So I think it's worth pursuing.
Okay. I guess, we've pretty much taken our time. I want to thank the panelists. I think we've learned a lot and raised more issues about the complexities between the immune system and how it can both be used to try to control cancer, but how at times it can be dysregulated and lead to autoimmune disease. I think this is going to continue to be obviously a really critical area. We've heard from some of the people who are leaders in these fields that have made the breakthroughs, and we hope a lot more breakthroughs are going to come from this field. So we thank you guys for joining us and for your attention, and we'll bring our session to a close. Thank you.
Thank you.